So you've spent literally weeks memorizing the structure of the DNA double helix. You know your adenine from your thymine, your cytosine from your guanine, right? You walk into your Cambridge Biology 9700 exam feeling, well, incredibly confident. You've done the reading, you've drawn the diagrams, the whole shebang. But then the results come back, and you're staring at a completely failed question. No way, right? Welcome to this explainer. Today, we're cracking the DNA base pairing framework, and we are going to fix the exact mistakes that are unfortunately bleeding your marks dry.
Take a look at this. 0 marks awarded. Candidate confused cause and effect. Ouch. That is a direct quote from the examiner's nightmare files. It's honestly the single most jarring, fatal mistake students make in this specific topic. You can know the biology perfectly, down to the last hydrogen bond. But if you flip the cause and effect when explaining complementary base pairing, your grade just flatlines. Literally, top-tier students lose their A-star right here. And it's not because they didn't know the science, it's because they didn't know how to structure their logic. And honestly, the stakes couldn't be higher for this one. Topic 6.1, DNA double helix structure and complementary base pairing, doesn't just pop up for a measly mark or two. No, it accounts for approximately 15% of the total marks in biology paper 2. 15%. To put that into perspective, that is a massive chunk of your final grade. It is literally the difference between passing and failing, or making that huge leap from a B to an A. So mastering this, it's not just some optional study session. It is the absolute foundation of your entire exam strategy.
Okay, let's dive into this with our core concepts, looking specifically at the traps of DNA structure.
You see, the syllabus dictates that everything here rests on precise definitions and causal relationships. But here's where students consistently stumble, evaluating evidence and drawing conclusions. When the exam asks you to handle experimental data regarding base pairing, like say interpreting percentages of nucleotides from a graph or a table, it's not just a memory test, it's a rigorous logic test. The assessment framework strictly prioritizes analytical depth over superficial breadth. Basically, you have to prove you can handle the data. If you think standard revision is enough to get by, well, read this specific warning straight from the Cambridge Principal Examiner report. Candidates consistently confuse key terminology, always define terms before using them in extended responses. This proves that ordinary revision, just reading the textbook, highlighting a bunch of facts and hoping for the best, completely fails. If you drop a word like purine, pyrimidine, or hydrogen bond in an extended answer without establishing its exact biological context first, the examiner absolutely cannot give you full credit. You must set that foundation.
Which brings up a pretty massive question. What is the hidden examiner clue that changes your answer completely? In just a few minutes, I'm going to show you a super subtle clue buried right there on the exam paper that dictates exactly how you should structure your response. Seriously, if you click away from this explainer now, you'll miss the exact trigger that turns a 0-mark answer into a full-mark answer. But wait, before you can spot that hidden clue, you need a very specific, unbreakable framework.
And that brings us to the high-mark answer framework, which is also known as the method mark strategy.
And this brilliantly illustrates how marks in Topic 6.1 actually work. They aren't just awarded for stating the final correct biological fact. No, they are specifically awarded for the systematic process. The framework requires you to systematically deconstruct the problem, solve it logically step by step, and then evaluate your findings. It's all about explicitly showing your scientific reasoning on the page. Here's how it goes. Step 1, identify the concept. Step 2, extract the evidence or data. Step 3, apply scientific reasoning. And Step 4, use precise examiner wording. This is the exact standardized sequence you need to build a top-band, evidence-based response. For instance, if you're handed a data table on DNA composition, you don't just jump straight to the conclusion. First, you state the concept of complementary base pairing. Second, you pull the exact percentages from the table as your hard evidence. Third, you logically explain the specific hydrogen bonding that causes those percentages. And finally, fourth, you wrap the whole thing up using the strict terminology the examiner expects.
All right, time for an interactive challenge. Let's see if you can spot the flawed answer.
Now, what's really interesting about this slide is the stark, undeniable contrast between these two approaches. I'm showing you two different ways to tackle a data interpretation question based on DNA-based percentages. Feel free to pause this explainer right now to see if you can spot exactly where the flawed answer on the left went wrong before I break it all down. Okay, ready? So look at the flawed answer. It says adenine binds with thymine, so they are similar amounts. Honestly, it's incredibly vague. It completely lacks any real data extraction. Now look at the improved top-band answer on the right. It perfectly applies our 4-step framework. It applies the concept by explicitly stating due to complementary base pairing. It uses the data by quoting A is 30% and T is 29.8%. And it provides crystal clear scientific reasoning by noting the two hydrogen bonds forming between specific purines and pyrimidines. The left side, that gets maybe 1 mark for luck. The right side secures absolutely every single available mark.
Let's move on to some top examiner warnings revealed.
So how do we make sure you're consistently writing answers like that top band example, especially under intense exam pressure? Well, we have to look at the exact pitfalls others have fallen into. Let's look at the areas where thousands of students drop marks every single year. These are pulled directly from the principal examiner reports because, well, we all know just how unforgiving Cambridge examiners can be. Here are the top 3 mistakes you need to mentally log right now. Number 1, confusing cause and effect. Do not say the stable double helix structure causes the base pairing rules. Actually, scratch that. It's the other way around. The chemical properties of the bases dictate the specific pairing, which then results in the structure. Number 2, forgetting to check units in terminology. Mixing up a nucleotide with a nitrogenous base will definitely cost you. And number 3, providing answers that are not specific to the given context or experimental data. To really lock this in, I highly recommend creating a first-letter mnemonic memory hack. Take the first letters of your absolute key terms, like purine, pyrimidine, hydrogen, bond, antiparallel, and string them into a memorable sentence. Having that instant recall is a total game changer when the clock is ticking.
So, the crucial point is, we have our framework down. But wait, here comes the diagnostic cliffhanger.
Understanding the raw biology of DNA is really only half the battle. Applying that 4-step method mark strategy is what actually earns you the grade. But there is a massive catch. Let's say you've memorized the framework perfectly. You extract the data from the graph, you define your terms flawlessly, you explain the hydrogen bonds beautifully, you walk out of that exam hall thinking you've absolutely nailed it. But then you get your paper back and you see a giant red 0. Why? Why does a biologically perfect describe answer get exactly 0 marks on an evaluate question? This is that hidden trap I mentioned earlier. Knowing the DNA framework is completely useless if you misinterpret the command words the examiner uses to trigger that framework. If you don't know the critical difference between describe, explain, and evaluate, you are actively throwing away your grade regardless of how well you actually know the biology.
You simply cannot afford to make that fatal mistake. And in our very next explainer, I'm going to break down the exact command words that decide your mark band. You absolutely must know what the examiner is secretly asking you to do. Subscribe to our YouTube channel, Ace the Exams, at Ace the Exams IGCSE EGC to excel in your Cambridge examinations. Watch the command words explainer next, and I'll see you there.